Okay. So, hello everybody. It's my great pleasure to welcome Claudia Redenbach as the speaker of our today's Lumer Lecture. Claudia is a professor at RPTU Kaiserslautern-Landau, and her main research focus is on stochastic geometry, so with a particular emphasis on random tessellations, spatial statistics, and applications of these mathematical methods to material science. So you see that there is a strong overlap with our working group 4 in the cost action mSPACE. And in today's talk, Claudia will give an overview about the methods recently developed by her and her team. And here we will also see a connection to modern methods from machine learning. So now we are looking forward to your presentation.
Yeah, thank you. And thanks a lot for the invitation to give this presentation. So I want to give you an overview of what we did in some recent and partly also not so recent projects. The main point is to put emphasis on the possibilities that you have to include stochastic geometry models in the analysis and description and also optimization of microstructures. I rather want to show a few different questions and different applications, so I usually will skip the technical details, but I'm happy to answer questions about that later. So we are talking about stochastic modeling of microstructures. Why are we doing this? Well, nowadays materials are highly optimized to different fields of application, and the microstructure is one key ingredient to tailor the performance of materials and also different physical behavior. Similar things apply to natural materials, where I mean things like bone, tissue, wood, or even polar ice. So also their particular structures would arise from geological processes due to forces acting on a bone, for instance. Also there, some kind of evolutionary optimization process is taking place. To get an understanding of how these things are related, one needs to characterize microstructures, and that means one has to observe them. And nowadays, various methods of 3D imaging are available, which make even very small-scale microstructures visible and allows us to quantitatively describe different microstructures characteristics. Why would we do that? Well, in first place, we could just find a statistical description of the microstructure. This way, we could compare microstructures. For instance, if we change parameters in the production process, we would want to know how that influences the resulting microstructure. Mechanical tests are often performed, and we would ask what happens to the structure after having applied some loading. Do cracks occur? Do displacements occur, for instance? And if you think of a more medical application, one could, for instance, ask how does a diseased or healthy tissue differ from each other? So that would be helpful for diagnostic questions. Once we have characterized the microstructure, we can fit a stochastic geometry model. So we could use a suitable model class and then also find parameters to match the observed structure as closely as possible. What are examples of those microstructures? If we start on the engineering side, we have worked quite a bit on foams, where we distinguish open foams here that are formed by a connected system of struts, or closed cell foams that consists of single cells that are completely separated from each other by thin cell walls. Fiber composites are widely applied nowadays, and here things like the fiber orientation distribution or also the length distribution would be important, for instance, for the mechanical properties. For porous media, you might be interested, for instance, in the permeability, if you use them as filtration media, for instance. And for concrete samples, civil engineers are interested in the grain system, because also that would have an influence on the performance of the concrete. Again, more on the biological side. Here we have two examples of CT scans of the blood vessel system in mouse livers. One is a healthy liver, the other is from a mouse suffering from fibrosis, and you visually already clearly see that there's a difference in the microstructure. Then we have the airways in the lung, where we were interested in mucus deposition, which is the green thing you see visualized here. And we also had studies with glaciologists, where we were studying the air pore system in polar ice cores, which at higher levels of depth would be such a connected system of pores, whereas if you go deeper in the ice core, it would then be a single system of separated, isolated bubbles. Where do we get our data from? Common imaging techniques in material science include scanning electron microscopy, which is very high resolution, but initially two-dimensional. So here we see an example of a non-woven fiber system imaged by SEM. And you see, since the material is porous, you can take a look into the pore system from the top. Another established imaging method, in particular in 3D, is microcomputed tomography. So here we see an example of a fiber composite. So here we have fibers in a polymer matrix, and in particular, fibers are nicely visible. So you could analyze, for instance, the fiber direction distribution. The difference in voxel sizes that you could achieve is in mu-CT, you would usually be in the range of around a micron, a little bit below. Probably for SEM, you rather have voxel sizes in the nanometer range. If you want to get a 3D using such a technique, you can use a serial sectioning technique, namely cutting your sample by a focused ion beam and iterating this with SEM imaging, where you could then slice-wise image your microstructure and stack that to also get a 3D image. Now, given such images, we want to characterize the microstructures, and we will see several tasks of how to do that. Why are we interested in that? We see here the kind of pipeline of what we call virtual material design. So on the left, you have a 3D image of a real sample, here a closed cell foam that has been imaged by CT and gives us an idea of how a material that is currently in use may look like. We could analyze the sample, we could decide on a certain stochastic geometry model, fitting everything together, finding model parameters. We would be able to now generate virtual realizations. So now this blue sample here is no longer real, but it has been computer simulated. And it could try to match the cell system that we see in the yellow example as close as possible. Now the question is, how does the cell system here influence the performance? So for instance, here we simulated a compression. So what happens if we sandwich our foam between two plates and apply pressure? So you see some estimated simulated displacement field here. And then the nice thing about having models is that you could do turns in changing the model parameters. You could make the cell size distribution more or less homogeneous. You could make the cell walls thicker or thinner. You could elongate your cells in a certain direction. And in each case, you could ask which influence that has on the resulting properties. So that means running parameter studies on the model allows to determine directions for optimization of materials, and compared to experimental testing, that's of course a comparably cheap and easy approach. So I want to start from that point. So and give you an example of virtual design of foam structures. So we have seen some examples already. Foams are made from various materials. So it could be polymer, metal, ceramics, for instance, and they are also used in a wide range of applications, from filtration to crash absorption, heat insulation. And you see here, there's a kind of a range from an open cell foams just consisting of the struts to a closed cell foams, but you also have hybrid versions where you have some open cells, but also some closed walls present. Foams are typically very nicely imaged by 3D and by a micro CT. They have a very high porosity. There's usually just one material present that you have to distinguish from air. And therefore, CT is a nice method to visualize and analyze the microstructure geometry here. The models of choice to get the virtual cell systems are Laguerre tessellations, so basically Voronoi tessellations and their generalizations. So here you see an example of a Voronoi tessellation. So the idea is you put points in space. Here is just a 2D example for visualization purposes. And then the cell of such a point here would consist of those points in space having that particular point as the nearest neighbor among all the black dots. If you look at this cell system, you see that the cells have very diverse sizes and shapes. You see rather small ones, rather big ones. You also see very small angles here, for instance. And that's not very consistent with the laws of foam formation, which rather suggests that the cells should have a regular, more roundish shape with almost equal edge lengths. So if you want to move into that direction, you could arrange your generator points more regularly in space. So here we see so-called hardcore point processes. So the hardcore point process, the points are not allowed to get arbitrarily close. And you see immediately the cells look much more regular. Now, if you also want to control the cell size distribution, you could go one step further and attach a weight to each point, here represented by these circles, with the idea being the larger the radius of the circle is, the bigger the sphere or the cell should grow. And this gives you another degree of freedom that could be used to describe not only the cell shape, but also the cell size distribution. So formally, what we would do is we take points xi, associate radii ri, so that's the radius of the spheres shown here. And then the cell of such a pair x and r would consist of all the points y that minimize this so-called power distance among all generators that we have. So this gives us the Laguerre tessellation, and this will be our foundation for foam modeling. How does the modeling chain look like? So initially here, for instance, we have an open cell foam. To describe the cells which are initially not really visible, we have to reconstruct them in the image using a watershed transform. So that happened here. Then we would fit a Laguerre tessellation to the observed cell system. We would take the edges of the tessellation cells visualized here, and then we would suitably dilate them to kind of attach material to the edges. There are various ingredients to such models. So one thing is the cell system. So we already discussed cell size distribution, cell shape distribution, regularity of these shapes. So for instance, which angles can show up? Should they be of equal size? Are very small angles allowed? Then if we look at the struts, here we see two examples of different cross-section shape. One is perfect cylinders with a circular cross-section. The others have a triangular cross-section, as you also see them very often in real foams, and clearly also the thickness of the struts can be modeled. In the right side here, we also introduced some closed walls. So that's such a hybrid foam with some open and some closed windows. And if you want to, you can also, for instance, play around with the anisotropy. So you, for instance, could elongate cells in particular directions, or you could play around with the orientation of these closed walls. We were using such models in a project where we wanted to establish a filter medium for cell chromatography. So the idea is you have a cell suspension, and maybe you have different types of cells, say good ones and bad ones, and you want to filter out the bad ones. And the idea would be is you take your filter medium, you somehow functionalize the surface in a way that the bad ones like to be attached to the surface, whereas the good ones wouldn't care and just continue their way through the medium. And then basically you pump your suspension through that filter medium, hoping to catch all the bad cells and having only the good ones left in your suspension. So here you see an illustration. So cells being these little red particles, and then hopefully they are caught somewhere on their way through the filter. What we did here was that we modeled the cell systems by Laguerre tessellations, and then we produced these foams or the filter media by 3D printing. So this is shown here. The challenge was that these are very delicate structures. So cell diameters are around 20 microns. So we decided on facet diameters on the range of 200 microns. And we needed very specialized 3D printing equipment done by colleagues here at RPTU. Here you see a picture of such printed foam samples, and you see a hair for comparison of how small these structures actually are. And well, when deciding which kind of model to actually use for printing, you would also have to ask how are the filtration properties related to the microstructure geometry. And this we investigated in a simulation study, at the moment only focusing on permeability. So in first place, there are no particles or cells present. We are just interested in the flow of a liquid through the material. So we looked at various model variations. We fixed the porosity to 95%. I mean, porosity would clearly have a very high influence on permeability. This we didn't want to study, but rather basically everything else. So we fixed also the same Laguerre tessellation. So kind of the frame of this filter medium would always be the same. What we did vary was the strut cross-section shape. So we had circular cross-section to get cylindrical struts, and then these triangular ones as you see them here. We used the original cell system and a cell system relaxed with Brache's surface evolver. The idea here is that in some studies we observed that Laguerre tessellations, even though being already more regular than some of the Voronoi examples that we have seen, tend to have too many small edges, and a surface evolver mimics steps of foam evolution, and it makes the cell shapes even more regular. And we saw that this makes them also closer to some foams that you really observe in practice. So in some cases, we found this relaxation to help to look more like the observed structures. In others, we actually do have rather irregular cell systems, then you rather use the Laguerre tessellation as is. And another thing that we did was we introduced these closed windows, the motivation basically being to provide some areas where the cells could settle down. And what we did was also vary the amount of such closed windows, trying to find some kind of compromise between high permeability, but then on the other hand, also surfaces to settle on. When it comes to selecting how to close these windows, we chose two protocols. One is isotropic, meaning every window was equally likely to be closed. And in the other situation, we only closed them orthogonal to the flow direction, with the motivation to basically put something in the way of the cells that are flowing through our medium. We generated 20 realizations per model variation, and we computed permeability using the GeoDict software. And our main idea was, can we find characteristics that explain the observed permeabilities? And motivated by a work by Westhoff and others, we chose tortuosity and constrictivity as such characteristics. Roughly speaking, tortuosity would measure how winding paths through the pore space are, whereas constrictivity is a measure for bottleneck effects that you have. So how small are closest parts that the cells or the flow would have to squeeze through. Interestingly, we could find one joint model for all cases considered, meaning overall strut shapes, coordinate directions, and closing mechanisms. We had a linear dependence initially between tortuosity and the Darcy numbers, which are a dimensionless version of permeability. And constrictivity could basically explain the difference between the two cross-section shapes, so circles and triangles that we used. So here is a visualization of the regression model. So here are the triangular shapes, here are the circular shapes, and the black frames are the isotropic models, no frames are the anisotropic ones. Then you have the X direction and the Z direction, which in the isotropic forms should be the same, in the anisotropic ones should show a difference. And the brightness indicates the percentage of closed windows. So basically what we see is if you stick to the same shape, then the more windows you close, the more the permeability goes down. That's not a surprise. Also, of course, in the anisotropic ones, the direction would then play a role. So if everything is closed in Z direction, the Z permeability goes down. And when it comes to the different shapes, these triangular struts tend to be kind of locally a bit thinner, meaning they extend more into the windows, making the space for the flow to squeeze through smaller. So therefore, this explains the difference here in the constrictivity, and with that also the permeability. Okay, so coming back to our picture here, now we have kind of by simulation investigated this part here. So if I give you a model, if I play around with the microstructure, how would that influence the properties? In the next example, I want to show you that model assumptions actually can already be used on the analysis step. So one example for that is a study that we did on detecting branching points. So again, starting with foams that we are already familiar with, you could consider such a foam as a multiply connected network of more or less cylindrical objects. If you think of, for instance, the blood vessel system that we saw earlier, there similar reasoning could apply. Locally, a blood vessel is something cylindrical, and every now and then they would branch and merge. So therefore, one could be interested in analyzing the connectivity. And here we developed an algorithm for detecting such branching points, shown by the little green dots here, based on the assumption that you have something locally cylindrical, and at the branching points where the cylinders are stuck together, the structure is slightly thicker than in the center. So the idea was then we would compute a skeleton of the structure showing in white here, and we would look at local thickness maxima along this skeleton, and this way detect the branching points. For the foams, that did allow us to cut such a foam into the different struts, giving us the opportunity to, for instance, study things like strut orientations or strut length distribution in more detail, and also use them directly in the model. As I mentioned, blood vessel systems form another example. So here we have synchrotron images of the mouse livers that you have seen earlier. So here we have two fibrosis models with different differences that you have in the blood vessel morphology. And we have a control group. So this is what I called the healthy mouse initially. And if you detect the branching points and look at the radius, so I said we look at locally the thickest structure, you see here in the healthy mouse, you have like an average radius, something around 3. If you look at the fibrosis cases, you see on one hand that the spread is getting larger, which is an indication of the morphology getting a bit more variable under fibrosis. And what you also see in particular in this case here is also the number of branching points clearly decreases. So, and that's an indication of change in morphology, which then would ultimately also influence the performance of the liver. And finally, we also used the same approach to analyze polar firn. So that's a kind of ice structure that we see in the upper layers of polar ice sheets. So in the transition from snow to this bubbly ice that we also saw earlier. So here we have lab CT scans provided by the Alfred Wegener Institute in Bremerhaven. And we analyzed the pore space. So what you see here in gray are air pores in the ice. And the observation is the deeper you go in an ice core, the more the porosity would go down. So this system, they start to split these different pores. So if you see look, these kind of peanut-shaped objects, basically the neck between the two thicker parts would split, and ultimately only isolated bubbles are left. So also here we applied our branching point detection, and we looked at different depth. So here, if it says back, that's a clashology unit for depth, which is roughly 0.5 meter. So this way we go down in the ice core, and what you see is the number of branching points goes down, indicating this splitting process going on. What we also did is we cut the samples into slices in the 3 coordinate directions and tracked the number of branching points over these slices. So X and Y, that's the surface of the ice, and Z is the depth, so going vertically into the ice core. And what you see is in X and Y, it's basically constant, whereas in Z, we see a large variability, which is explained by the fact that in ice, you also observe seasonal layers. So depending on climate and weather conditions, the ice may show different behavior, which also influences these processes, these splitting and bubble formation processes in the pore space. And that's clearly visible here. So now you see we could use the very same assumption and the very same algorithm to structures coming from very different fields of application, basically abstracting from what exactly things are and purely concentrating on what they are geometrically. So therefore, from this more abstract mathematical point of view, that gives you the freedom to transfer things from one field to another that initially probably are thought to have nothing in common. As a second application example, I want to show you how to use model assumptions in the analysis of fiber systems, and particular focus will be an estimation of the length distribution. So if you think of glass fiber composites, for instance, there it could happen that during processing the different fibers break, meaning that initially you wouldn't know what the length distribution in a composite is. An established procedure for length estimation is an etching test, meaning you would burn away the polymer. This way, fibers are getting loose and you could disentangle them and measure their length, for instance, under a microscope. However, also in that process, additional fiber breakage can be introduced. So what we are investigating is how we could estimate fiber length distribution from CT data, which initially requires a segmentation of single fibers. That already is a challenge, but in addition, we see here that the estimation is hampered by edge effects. So you see a lot of fibers are actually cut off by the image boundary. So we wouldn't know how long they actually are. We only know how much of them we see. A classical approach for removing such edge effects is an approach by weighting, which is called a mild slant to Joule, or also Horvitz-Thompson correction. So the idea is you only keep fibers that are observed completely. This way, you introduce a bias because longer fibers are more likely to be cut by the edge of the window, meaning in the remaining ones, you have a bias towards short fibers. And weighting this with the 1 over the probability of being observed kind of can correct this particular bias. However, you see here in such an example, quite a lot of fibers can be cut and all the information for them is lost to the analysis. So the question was, can we take the information from censored fibers into account? And we found some approach by a Swedish group using an EM algorithm in 2D to perform such an estimation. And then a PhD student generalized that to 3D and the question of fiber length estimation. It is a parametric approach where we assume that the fiber length distribution is log normal. And we also assume that the directional distribution of the fibers is known. That's really not a problem in general because there are powerful methods available to estimate that distribution from data. What we could do now is assume we have a single fiber segmentation. We would look at the visible length. So this is the length of the fibers that we see in the image. It's not the actual length that is denoted by L without the tilde. So for fibers that hit the edge of the window, it would mean L tilde we see, and L is what is in the window plus what is outside the window. And then if we want to estimate the parameters, for instance, if we wanted to estimate the mean of the logs, then the idea would be to do it this way, that you would take a sample mean of the expected log of the actual length given the observed length, because that's the only thing that we have. How do we do that? You see here, we would need the distribution of L given L tilde. Well, we could use Bayes' law to say, well, that's the distribution of L tilde given L times P of L over P of L tilde. And the things on the right-hand side are known. So PL is our log normal law, and the probability of L tilde given L can be computed by geometric considerations. So you could ask, if I have a certain fiber of a certain length, how likely is it that I see L tilde of that thing in my window? So that's kind of doable computations to figure these densities out. However, there are different cases have to be considered, namely this density depends on the one hand on the sampling rule. So how do I decide which fiber to measure, and also the fiber direction distribution. And then we would sample from that density here to get a Monte Carlo estimate of this expectation. Theoretically, you can also prove that this AM estimator is consistent and asymptotically normally distributed. Empirically, we could show that we get acceptable results already if the window edge length is about as long as the mean fiber length. But we are still working on a more thoroughly validation of that approach. Now I said you need a single fiber segmentation to be able to run that protocol. If you look at an image like this, so very high fiber density, it's hardly possible to get that segmented. So in particular, in practice, if you want to fit in the length of the fibers, you have to make a compromise regarding resolution, and then spaces between the fibers may be poorly resolved. For fibers of that kind here, so very low density and high resolution, that's doable, but here it can get hard. We thought of some alternative. So we asked the question, well, if we just were able to see endpoints of fibers, would that help us to estimate the fiber length distribution? So, and the answer is yes, that's possible, but under very heavy model assumptions. Namely, if we assume that the fiber center point is a stationary Poisson process, which basically implies that the centers are thrown independently of each other with a given length and a given direction distribution, then we can use the fact that if we just look at the endpoints, so just these points shown here, without taking into account how the fibers were initially collected, then we can get a non-parametric estimation of the fiber length distribution. How is that done? There is a classical spatial statistics summary statistic called Ripley's K function. And for such a process where you have a stationary Poisson process for initial points and then clusters, here the clusters being the two fiber endpoints given by some fixed distribution and being drawn IID, then this Ripley's K function can be computed explicitly, and it does depend on the fiber length distribution F. And it's very well known how to estimate K, and then you just have to solve the formula that you get for the F. It's kind of surprising that just from such a pattern where no fiber information is left, you could still see the length distribution. But in a simulation study, we could show that actually for a large number of fibers, it worked well. So here in circles, you see the actual distribution. So now we get a non-parametric estimate, so basically a histogram. The initial estimate is here the little stars. This can be improved if we take into account the direction distribution of the fibers. So, well, from the academic point of view, that's quite nice. And it shows how a high amount of loss of information can be compensated by very heavy model assumptions, and then still you can reproduce information. Unfortunately, on the practical side, we don't believe this to be of too much value because real fiber systems where the fibers are non-overlapping are relatively far away from that Poisson assumption. So therefore, whether this relation between the length distribution and the K function still holds is highly questionable. But for illustration purposes, I think this is a very nice example. Okay, so now we have discussed that our model assumptions would help us in the analysis of the microstructures. Now, finally, I want to go to the image processing and show that models are also valuable there. I mean, typically, the images that you get from SEM or from CT are gray value images, and what you have to do is you have to segment them. So here are some examples. So you have gray value images. In this case, it's CT scans. And for instance, here we see the strut system of a foam. Here we see a fiber system. Here we see some grains. And if we want to analyze them, we have to distinguish them from the pore space or from the matrix. Ideally, and that's possible here, this can be done by simple thresholding, meaning you just set a threshold and say everything that is brighter than a certain value is the component of interest. So here you clearly see this is easily possible. But I will show you an example where it's not that simple. Where did we do that? Well, we had a project called Danobi, which was about segmenting cracks in concrete. And such cracks occur due to the application of mechanical load or corrosion, or sometimes also chemical reactions. There are possible tasks. One thing could be that we ask, is there a crack in the image at all? Or what we are interested in, crack segmentation. So find all pixels that belong to the crack. Here we clearly see that simple thresholding does not do the job because there are other things present here, for instance, like air pores that share the same gray value distribution as the cracks, and therefore just by thresholding we would get everything. But we are just interested in that crack here. Why do we do that? Well, recently we established a large computed tomography device at RPTU in Kaiserslautern, which is called Gulliver. And it's designed for scanning large samples under load. So here you see an example. So we can scan concrete beams up to 6 meters length and subsequently apply some load here and, for instance, scan these beams under load and this way visualize the evolution of crack systems, for instance, in the concrete. When we were starting to work on that, we got an overview of the literature, so which methods are on the market. And there are different groups. One is basically derivative-based filters, where we could ask in which direction do the gray values change. There are connectivity-based methods, rather saying a crack is a long, dark thing that I could track through the image. And then, of course, nowadays, machine learning and deep learning methods are getting more and more interest in image segmentation. So we also looked at random forest segmentation and neural networks. So here the 3D UNet. Well, why would one consider classical image processing at all? Nowadays, machine learning is often considered the method of choice. Well, one point is classical methods do not require training. They are also kind of based on model assumptions on the shape to be detected. So for instance, for a crack, you could say it's a locally planar, thin, dark structure in the image. And that's basically the information that you base your segmentation algorithm on. However, what usually is required is to choose a scale parameter. So if you want to detect this very thin crack here or that very rather thick one, you would have to work with different scales to find everything. So we will come back to that. In our comparison study that we refer to here, we found that the Hessian-based percolation was the best classical method, and among the machine learning method, UNet gave the best results. Now we could ask, how is it possible to actually say that one method is better than the other? We need to be able to assess the accuracy. And also for the machine learning methods, we need some training data, so we need annotations. Nowadays, manual annotation is still often done, but the quality of the annotation is hard to evaluate and it's very time-consuming. So what we did is we used the stochastic model for a ground truth crack generation. Then we superimposed such a synthetic crack here with a real image of concrete without a crack. And then we had a perfectly annotated pair of a gray value image looking like a CT scan of an image with a crack, or for concrete with a crack, and the mask for the ground truth annotation. Regarding the gray value distributions, here we made use of the fact that we could look at the pores. So the gray value distribution in the pores was used to decide on the gray value distribution in the crack. How did we do that? How did we simulate the cracks? It's also based on a Voronoi tessellation. What we did here is we selected a subset of the facet system by solving a minimal surface problem in the tessellation. Starting from such an initial curve, we found a closed surface, which is then our crack shape. We also published a data set. So if you're interested in using such images, there is VoroCrack 3D, which is a data set where we have simulated cracks of different widths, different variability of thickness, branching, different amount of noise added, also different concrete backgrounds that now could be used for such segmentation studies. And that's available on Zenodo. Furthermore, I also mentioned these multi-scale cracks. So we already discussed that probably you should choose different scale parameters. If you do that, so if you run your method multiple times with different scale parameters to detect either the thin or the thick crack, that of course increases the runtime. So therefore, we were interested in a method that could segment multi-scale cracks in a single run. And this was indeed possible by a modification of the network that we are using. Namely, we replaced the convolutions used in the UNet by a so-called Ries transform, which is given here. And then we could benefit from the fact that Rieström's forms are scale equivariant. And that means that we get a scale invariant segmentation. So if we take a network trained on fixed crack thickness, so for instance, 3 pixel thick cracks, if we use a standard UNet, it would basically just find the boundary of the cracks because it wouldn't expect such thick, dark regions and wouldn't consider them as cracks. ResNet, on the other hand, does have the capability to generalize, so that would find cracks of very varying thickness, even if trained on fixed scales. The next step will be to get our methods more robust, because you see here, concrete is not the same as concrete. There is a huge variability of ingredients yeah, also morphologies. So we will work on getting the method robust to changes of the concrete. A final example that also considers SEM imaging. So if you scan such a porous sample, I already mentioned you can look into the pore space, and it can be very hard to determine what is the uppermost layer of your material. So here the question is, how can we get it segmented? So if I gave you a pen and asked you to manually annotate the uppermost layers, I think you agree that this would be rather challenging. So also here we wanted to use a machine learning approach, and here the ground truth was obtained from FibSEM simulation developed in the PhD of Torben Prill. So here the idea is you take a virtual microstructure, so using one of our models, and then you would simulate the physics of electron-matter interaction that happens during SEM scanning. There is a library called Monsel2 that can be used to simulate the electron path, and Torben developed various methods for accelerating the simulation. Still, the simulation takes several hours for reasonably sized images. So therefore, we worked on a deep learning acceleration. So the idea is you have a microstructure represented by balls, cylinders, or cubes. And then we have a ResNet surrogate model that is trained on data simulated by Torben's method. The main point here was to get the kind of a nice representation of the microstructure. So here height fields and normal maps turned out to be very useful. And this way we can simulate a single slice in 20 milliseconds. And here you see some examples. So on the left, we have a physically correct simulation. Here we have the result of the machine learning method. They look very similar. What we do see is that the machine learning model does not produce noise. This we have to add afterwards. And here you see the residuals. So there are some tiny things at the edges. Here there's one pore where something is lost, but otherwise it looks very convincing. So to conclude, I have shown you that quantitative image analysis yields important information on microstructure characteristics. And in particular, models from stochastic geometry can be used for various purposes. In some of our studies, they were used as prior information to be incorporated in the image processing and analysis. We have used them to generate ground truth data for method evaluation and to get training data for machine learning approaches. We are able to reproduce characteristics of observed microstructures. And using our models, you can produce many large samples with altered microstructure in relatively small time compared to experimental studies. So in combination with numerical simulations, this allows for the investigation of microstructure property relations with a much reduced amount of effort compared to experiments. So this really paves the way for a virtual design and optimization of materials. So, and with that, I thank you for your attention, and I'm looking forward to your questions.
Thank you very much, Claudia, for this nice overview. Are there any questions in the audience? Daniela?
Thank you. Thank you, Claudia, for such a nice contribution. I have one obligatory question that I have to ask you with my background, of course. So you mentioned that the choice of the underlying point process for the simulations is essential. And a lot of times Poisson point process is just not sufficiently flexible or too messy, let's say, not so regular. On the other hand, hardcore point processes are regular in some sense, but also not that flexible because of the hardcore. So I need to ask you if you, by any chance, tried some hyperuniform point processes, for example.
We haven't tried using them yet. It would actually, I mean, given it's a rather recent development, I think this is something that one could investigate. Yeah, so usually so far, I mean, the Poisson is usually the only thing that is somehow analytically tractable, whereas from the realistic point of view, well, things in materials do interact. So therefore, the Poisson assumption is usually not very realistic. So there you always face a trade-off between analytical tractability. So in that fibre example, you saw that it can be very powerful to have these assumptions. But when it comes to realistic examples and performance and practice, one usually has to go to
as you said, I mean, there is a certain trade-off always.
Yeah.
But there are a lot of now new results in the hyperuniform sector. And some of these characteristics are actually already tractable. For example, the K function of one special case, let's say, is tractable. And this is like a lattice if you perturb by Gaussian random vectors. Then the K function itself is tractable. It could be explicitly written down. So maybe this could be of any interest in the future. I don't know how to apply it then, but
yeah, it's something one should definitely look into. Yeah.
All right.
Thank you.
All right. Thank you. Thank you.
Delio, you also have a question.
Yeah, thank you very much for your very interesting talk. I was wondering, okay, so first of all, I have a theoretical question. So at a certain point you were studying or you were discussing the role of Riesz transform. And to me, as a person from a totally different background, this was a bit surprising. So which properties of Riesz transform specifically are you using in your modeling? But apart from this, more generally, I found the part and section on forms particularly interesting. So have you ever seen in your research something where the shape of a form is influenced by the physical activity ongoing there? Or maybe if you can think of more general porous media, if you have transport, if you have some slow flow, you are modeling by a Stokes equation, this might also apply some pressure on the structure, for instance. So do you observe change in morphology of your structure? Is this something that in your community is used? Because in other groups that appear in this cost action, there are people working on so-called co-evolving graphs. So this is just a 0 or one-dimensional model, but still you have this models where a network changes its shape, its topology under a physical process or along a physical process. Do you ever observe or model similar phenomena in your research?
Well, if you stick to liquid foams, so soap froth, or if you think of the foams you have on your bath or your beer, you can see evolution, right? The bubbles are kind of changing shape and size and are merging. And so their Plateau's laws are valid. So there is a certain assumption about how the bubbles should look like to have a stable configuration. Now, if we look into solid foams, we observe that there the shapes may vary quite a bit, probably because foams are frozen to the solid state earlier or later in such a process. So I mentioned this surface evolver technique. So there the idea is that you would mimic what happens in these soap froth, and I said it generates generally more regular, more roundish cell shapes, and also a much more homogeneous edge length distribution. And we used that for several foams. And for instance, we had an open metal foam where that helped a lot. So that seemed to be close to this nice surface area minimization configuration. So there, moving from the pure Laguerre tessellation to the surface evolver gave us a result that looked much more than the data. We also had a closed polymer foam where it didn't help at all. So that looked way more irregular. And there the surface evolver went too much in the regularization direction. So therefore you should look at your sample and yeah, decide which of these steps you actually need.
Okay, thank you.
And I think you were asking a question about the Ries transform.
That's
so there the main point was the scale equivariance. So if you use a standard convolutional neural network, you would work with fixed window like convolutions, and these kind of have to match to the, or it would influence on which scale you see features in your image. And in the Ries transform, you have such an unbounded and scale equivariant structure. So in this case, it kind of would see the features no matter at which scale they occur. So it would see the crack kind of no matter how much you zoom in or not.
Okay, thank you.
In your talk, you have also mentioned application to biological materials like livers or lungs. And so in these biological materials, typically they also arise non-stationary or non-isotropic effects. And is this also on the scale you are observing? Because then the tools for statistically analyzing these things might differ from classical stationary settings.
Yeah, that's really something that we did observe. And yeah, there you have to use a highly localized way of representing it. If you think of the lung, for instance, these airway systems that we saw, they rather look like trees, right? You have the big branches, and then it's getting finer and finer. Clearly, kind of averaging that over the lung or something doesn't make too much sense. And I mean, here also the doctors were interested in how that goes or how it develops as you go deeper into the lung. So therefore, in this case, for instance, we decided to follow the kind of the skeleton of these airway branches and then analyze it slice-wise and visualize on cross-sectional slices how severe these mucous obstructions were.
Maybe one further question regarding your fiber-based materials. So is it also possible when measuring the length and you have fibers where you do not observe them completely, to doing something like a maximum likelihood estimation with missing data? Like, for example, when you're doing maximum likelihood estimation for lifetimes, and you also do not know where there is still some living species. So then you can also use for one part you use the density, and for the other one you use the distribution function in the likelihood. Can you also apply such methods, or are your methods advantages in the sense that you also took the spatial position of the fibers into account?
Well, in the estimation, we don't use the position, but just the sample. So what we would put in the estimator is the observed length, so these L tildes, plus the information if they are censored or not. So for fibers where we know that, I mean, we can see in the image if they hit the boundary. So for fibers where we know that this is the actual length, we use that information. And for fibers where we see that they are censored, we take this approach to saying what is the expected actual length given this seen part. So, and yeah, I mean, as it's an EM algorithm, it kind of the likelihood function, of course, plays a role there. So you have to compute this density that you see there. The spatial placement does play a role when it comes to computing these probabilities. What is the visible length given the actual length? Because then you have to check where you could place them such that you see exactly L tilde.
Thank you. Okay, so if there are no further questions, then I would like to thank you again for giving this nice presentation to us.
Thank you. It was a pleasure.
Thank you very much.
Okay, yeah. Then I think we would close this Lumer lecture for today, and thank you all for participating.
Yes, maybe I can just add that. So first of all, thank you again, Claudia. And then let me just mention that the next Lumer lecture will take place on September 8th. It will be given by Barbara Tinthammer from Bonn, but you will get more information in due time. So see you at latest then, and otherwise, I wish you a nice summer, everyone. Bye.
Bye.
Thank you, and bye, everyone.
Thank you, bye.